This is a second webinar in a series of webinars we started this past May on special topics in cell isolation. We've been holding these types of webinars since November 2020 for human islet isolation, but I thought it was worthwhile to get some special topics discussion as well. And we had a very good first seminar from Rafe Wittek on the use of collagenase to isolate at high yields of human hepatocytes. So today I'm very honored to introduce Stu Williams, who, as you've seen from his bio, has extensive experience. And just prior to the call, I mean, he started isolating cells from lipoaspirate back in 1970. And so the great thing about him is that you can read about his history, but I met Stu back in 1996, about 1996, when we invited him to Indianapolis to talk about use of collagenase to isolate endothelial cells from human lipoaspirate. What he was trying to do is cell sorting of vascular synthetic grafts, and he actually has a patent on that device that he worked with Becton Dickinson on. So he's got extensive experience in this field. He also went as far, his interest in collagenase, I should say, went as far as him working with Harold Van Wart and subsequently writing a paper in Cell Transplant around 1995, I think, about trying to improve the quality of collagenase that was used in cell isolation. So without any further ado, I will turn it over to Stu to give the presentation. Thanks, Stu.
And thank you, Bob. It's a real pleasure to go back in history and talk a little bit about this whole process of isolating cells from fat. And as you see from the title, it's the 60-year anniversary of Marty Rodbell's original publication on how to isolate cells from fat. You see all my academic affiliations over the years. I'm now retired from academics. I still hold an adjunct position at the University of Louisville, but my whole science started in the top left. I was a third grader at Lombardy Elementary School in Wilmington, Delaware, where I was catching fireflies for the DuPont company, and they were isolating luciferase. And it was 40 years later, I used luciferase to trace SVF. Little did I know, but to used it to trace SVF injected intravenously in a rat model of distribution of cells. This is my financial disclosures. I hold a fair number of U.S. patents in the field of regenerative medicine, most of them in the area of fat-derived cells and how to isolate, equipment necessary. And here are all the companies and groups who have supported my efforts over the years. This is where it all started. Marty Rodbell, 1964, February. This was published in the Journal of Biological Chemistry. And the history moving forward of this whole field, it is amazing the number of different journals that isolation methods have been published in. And this has actually led to a little bit of the difficulty in tracing some of these methodologies because the biological chemists do not always read the obesity journals, and vice versa is the same. But Marty started the whole thing in 64. He'd been working on ways of trying to isolate cells from fat. And he came up with this novel collagenase that he got from Charlie Worthington in order to do this work. Marty got the Nobel Prize in 1994, and in the opening remarks of his Nobel lecture, he says he uncorked the means of freeing adipocytes from their tissue matrix by collagenase treatment. And then his seminal discovery was that insulin would stimulate glucose uptake by these fat cells. So a cell actually is an entity. Marty's somewhat the father of cell biology. has a brain and can actually work. And in his autobiography that was associated with his Nobel Prize, he went a little further to describe that following the digestion of adipose tissue with collagenase, and he specifically stated it's not a pure enzyme, it's an impure preparation that contains many proteases, including collagenase, that fat cells floated to the top. And I had the opportunity to talk to Marty several times in the late 70s, early 80s, while he was still at NIH in Bethesda. And he said that the cells that end up in the pellet are mostly vascular cells. So even at that point, he knew that the appropriate collagenase digestion resulted in a stromal vascular fraction that was predominantly endothelial cells. And this is pictures from his original paper and the basic method that he utilized. Taking rat fat, he used female rats and parametrial fat, the fat around the uterus, light mincing, enzymatic digestion. He got the Worthington collagenase, which is a clostridial collagenase. He had previously been using a clostridial Welchie strain of collagenase that was not, got that from a investigator by the name of Bidwell from England. It was not that effective at digesting the fat, but he used Charlie Worthington's collagenase that worked extremely well. He used an Erlenmeyer flask, a Dubanoff shaking water bath, 60-minute digestion, centrifuge it, and the fat cells go to the top. That's figure A. The vascular cells that he called the stromal vascular cell pellet is shown in C. And I did have the opportunity when I was doing my PhD and then also my postdoc to talk about that. In the pellet, does he believe that it's predominantly endothelial cells, predominantly vascular cells? And he basically said, yeah, that's the way he saw it, which was important to me because I was studying the pellet of cells. For any quality people in the audience, look away. This was the laboratory notebook from Marty Rodbell's work showing that GTP was the most active molecule in this process that he called signal transduction. It was not ATP, it was GTP, which went on to allow him to discover G proteins, and with Gilman, they got the Nobel Prize for this work. He actually switched from fat cells to liver cells and liver membrane ghosts, as he called them to do most of his work. And then there was a second paper in this series that did involve Marty Rodbell. And this was the paper by Wagner from 1972. It's work that Roger did in the late 60s while he was doing his postdoc in Russ Barnett's laboratory. Russ at Yale is the father of use of glutaraldehyde for electron microscopy. Mark Batinsky, a pathologist at Yale, knew Marty Rodbell extremely well because Mark was the one who discovered that light activated a G protein in our rod outer segments, which led to the discovery of how signal transduction from light to the brain takes place. So Marty and Mark spent a lot of time talking together. Roger took those conversations, took the pellet of the initial Rodbell digestion, studied it, and found two interesting things. One, adenyl cyclase could be found there, and two, there was micropinocytic activity. I ended up doing my PhD in Roger's laboratory at the University of Delaware, and I focused on this micropinocytosis. But it was Roger who was the first one who evaluated this pellet of cells. He called them capillaries, and he used the exact same method that Rodbell used, mincing of fat. Roger used epididymal fat, male rats, the Worthington collagenase, Dubanoff shaking water bath, centrifugation, etc. But he put in one simple modification. He put a filter in that captured the endothelial cell microvascular fragments, washed it, and then he studied what stuck to the filter, which were all of these blood vessels. And Roger spent a significant part of his career studying these capillary endothelial cells that were present there. Whoop. This is from 1972, Roger's original drawing. I was in his laboratory, and he gifted this to me while I was in graduate school. This is how he did the isolation of microvascular fragments, and it's still done today, capturing these fragments from the isolation of fat. And then it was 1981, work that I did as part of my PhD, where I basically did the exact same Rodbell isolation, the Wagner isolation, but I didn't use the filter. What I studied was that final pellet of cells that I called capillary endothelial cells, and did my PhD on this process. Same exact procedure that Rodbell used. Now, one of the things that I've done since I retired from both industry as well as my academic positions is about 8 months ago, I did a complete literature search, which I'm still in the midst of adipose cell isolation. That was my first PubMed search, but I have added on to that using a variety of different terms, EndNote, the Patent Office, Protocols IO, dissertations, to really find out what's happened since Rodbell did that original study. How are people isolating these cells? One of the things I've spent time doing is really researching the embryology of this, how we get to fat, and then also a significant amount of review that I've done on how we derive this term stem cells from fat and the infamous mesenchymal stem cells. I'm not going to talk a lot about the stem cells in this presentation. I'm setting up with Bob a second presentation where I'll delve more into how the stem cells are derived. I'm going to focus most of this presentation on how in the world did we get to where we are today in this enzymatic isolation of the stromal vascular fraction. And in this very busy slide, this video will be available so you can go back to some of these slides where I don't cover everything. I want to bring out 2 or 3 major elements. One, the little yellow dots are the progression of how we review the literature and how we see the progression of technology. In '63, Eugene Garfield started the Science Citation Index, and he started the process of evaluating publications, assigning keywords to those publications that could then be searched based upon the keywords. This was both incredibly helpful as well as problematic because investigators' keywords did not always capture everything that was critical in that paper as far as discovery of the process. And then you can see all those yellow dots moving through to, I think one of the latest things that I've been using a lot more is Notebook LM, a Google product, using AI in order to basically tease out how different technologies have moved ahead. There are a lot of different things. It's an election year, so I will point out for everybody to remember, in 1997, Al Gore made Medline free to the scientific community, so you didn't have to pay for it. The thing that's not written here is in the earliest days, and we don't do this as much anymore, when a paper came out that we saw in Science Citation Index, we would just call that investigator on the phone to find out all the hidden truths that were going on, what they didn't put into their methods section in order to move the technology forward. The second point in this is following Rodbell's publication of that enzymatic cell isolation method, he started studying adipocytes, and that continues on. That's the green. Uncultured SVF and adipocytes. These are the uncultured adipocytes, brown fat, white fat, all sorts of different fats. And that continues on until today. But there are other cell types that have been identified within SVF. One is the endothelial cells, and I can use my pointer. In '72, Wagner gave us, there are lots of endothelial cells in fat. That has continued in a lot of ways, the study of the endothelial cells of fat. Microvascular fragments has continued to be studied. Pre-adipocytes, the groundbreaking paper by Faust in 1978 that essentially gave us the concept that fat is turning over in the body. Before I learned in my classes, graduate classes, that you're born with all the adipocytes you're ever going to have, and you either get fat from them getting large or thin from the fat getting small. Faust disproved that and said there are pre-adipocytes, and pre-adipocytes, which is the first true stem cell in the stromal vascular fraction, were studied from 78 on. And then adipose stem cells, they really were developed, studied in the 1980s. And you see on the right, these are all the journals and areas where this was studied, and really a lack of interaction of investigators. Those who are studying adipocyte function did not spend a whole lot of time talking to people in the endothelial cell world. So the obesity and the vascular surgeons didn't often cross-reference each other in their papers. This has led to some interesting things that I've picked up over the past year studying all this. And one of the most interesting things is Rodbell in the beginning called that pellet the stromal vascular cells. And then you see on the right side of this, these are all of the initial investigators who used a different term. So Poznansky, Wagner, Van, Bjorntorp, myself, Hauner, we all called the cells in the stromal vascular fraction by a different name. And this has been unfortunate in some ways because it's all stromal vascular fraction. And I'll also make one of my first controversial statements. The only way to get a complete stromal vascular fraction is using enzymatic digestion. Non-enzymatic methods only produce in this pellet a stromal fraction. You have very few, if any, of the original endothelial cells that exist in fat in that fraction. All of these investigators used enzymatic digestion, and they all called the cells in the pellet, exact same pellet, by a different name. So one of the things that, as a takeaway, is I hope we start calling that pellet what it is after primary enzymatic digestion, and that is the stromal vascular fraction. Now, this is a individual economist that I have read many of his books, and this is one of his most famous quotes to me, which is, if I had to reduce my message to management to a few words, I'd say it all had to do with reducing variation. And this is the variability in the methods for the isolation of the stromal vascular fraction, which you see about 2/3 of the way to the right in this whole process, the primary cell isolate, SVF. And beyond that is the culture of these cells and all of the different variabilities in the culture of cells to finally lead to, I use the term cell line. In some people's work, this is the mesenchymal stem cells. In other people, it's the adipose stem cells. In other people, it's just cultured stromal vascular fraction. I'm going to go through some of these variabilities that all lead to optimal or suboptimal isolation of the stromal vascular fraction. All of these variations are now provided to you next to Rodbell's original procedure. And for the most part, the process of cell isolation of cells from fat in 2024 is basically the same. It's fat. It goes through different forms of storage, washing, and mincing. Enzymes, you'll see there are a lot of new enzymes that are essentially the same as the Worthington enzymes in many ways, agitation systems. And then this introduction of filters has taken place in this field. Sometimes the digest is filtered before it's centrifuged. Sometimes it's filtered after it's centrifuged. Different forms of the final product based upon the filtration and centrifugation speeds, etc. And then one final little filter that's almost hidden here, and many times it's hidden from people who do these studies, is a filtration that takes place prior to characterization of SVF and other cell types utilizing cytometry, fluorescence activated cell scanning or cytometry. Many investigators send their SVF off for characterization to the FACS laboratory, and often don't know that most of these laboratories technicians use a 20 to 40 micron filter, and they will filter out a lot of the most important cells in this product. I'm going to go through a lot of these variables. Just the variability, time of digestion, 4 minutes to 24 hours is the variability in the time of digestion of these cells or of this fat. Now, how's it all work out over the years as far as how good are we at getting SVF cells out of fat? In the middle, you'll see an orange line here. This is the theoretical based upon in situ cell quantitation. This is the number of stromal vascular fraction cells that can be found in a cc of fat. And it's around 3.5 to 4 million cells per gram of fat. So if it was 100% efficient, you would get somewhere around 3.5 to 4 million SVF cells per gram of fat. Here are all of the investigators where they have provided a evaluation, a quantitation of cell number per gram of fat. And you see, for the most part, most of these fall below. Rodbell utilized DNA evaluation and determined there were 11 million SVF cells per gram of fat. I talked to him about this in those early days when I was just learning the procedure, saying that I should expect 11 million cells. His DNA measurement and evaluation techniques had a lot of variability in them at this point. So this is, I would say, a spurious high. And there is out there in the literature, someone found 5 trillion SVF cells per gram of fat. This is instrumentation error. If you want to maximize your yield of SVF, that's the instrument that you need to use. And unfortunately, in this paper, they don't say what instrument they use to get this 5 trillion cells. That's a spurious number. But you see, in general, people are getting somewhere around, I would say, 10,000 cells per gram of fat. Relatively low number for the most part. They filtered the endothelial cells out up to a paper in 2011 where they got very close to 100%, what I'd call 100% efficiency. So there still is variability in literature. All right, second controversial slide that I'm putting up that in the second webinar I'll spend a little bit more time on. Where do cells come from in adipose tissue? I call it a model, and there's a very famous paper by George Box on essentially all models are wrong, but some are useful. But here is the model, my accepted model that I utilize to explain to people where the cells in fat come from, and it gives you an inkling of what I believe the true stem cell is in fat. These cells are derived from predominantly mesodermal cells. There is some evidence in the literature that neuroectoderm cells can give rise to fat. Goes through hemangioblasts, blood islands. Microvascular networks are formed due to a process known as vasculogenesis, and a subsequent angiogenic process can take place where there are a group of cells at the formation of this angiogenic sprout, which are known as the tip cells. These are the cells that, from my review of the literature and some of my own work, are the true stem cells in fat. I'll show you in a second where I don't believe parasites actually exist to any extent in microvascularized fat. It's these tip cells that in most of the literature review that I've done are the true stem cells in fat. They can give rise to smooth muscle cells. They can give rise to new microcirculation, hemogenic endothelial cells. These are endothelium that can give rise to a variety of hematopoietic cells, pre-adipocytes that give rise to pre-adipocytes to adipocytes, hematopoietic stem cells, lymphatic endothelium. The fat is innervated through a sympathetic nervous system, and there are circulating cells that do diapedese and take residence. So when you start looking at fat, Here is where all the cells come from. It's a complex, heterogeneous population of cells. So a little bit on enzymatic digestion. So you're going to go all the way back to my PhD dissertation, an fluorescent image of the cells surrounding fat cells in a piece of rat epididymal fat. The nuclei, if you take a close look using just straight light microscopy histology, you'll see that almost all of these nuclei are endothelial cell nuclei. No true evidence of parasites here. Electron microscopy, again from my dissertation, there are some cells that do appear to, segments of them arise or are found in the extracellular matrix surrounding these capillaries. But for the most part, it's capillary endothelial cells, continuous capillaries associated with the fat cells. These are scanning EMs utilizing a partial, what we call microdissection. In non-enzymatically treated fat, you will see the endothelial cell structure still bound by the extracellular matrix. So any non-enzymatic method for isolation of micronized fat, there are all sorts of different terms for this, the endothelial cells will not be released. It's only by enzymatic digestion that you see the release of these endothelial cells. And this is another example or rationale why I say that unless you're using enzymes to digest the fat, you are not getting a stromal vascular fraction. Non-enzymatic methods only provide a stromal fraction. You have a very low concentration of endothelial cells. And then in the literature, what are the cells found in fat? And it is all over the map as far as the percentage distribution of cells in stromal vascular fraction, utilizing a variety of different techniques. I think we all agree that these are the cells found in the stromal vascular fraction, but their relative distribution, you'll see some places in the literature, fat SVF contains no endothelial cells. Some 100% of the cells in the stromal vascular fraction are fat. You see the same thing for pre-adipocytes, ASCs. I think we can all agree that it's a heterogeneous population as far as the distribution of cells in the stromal vascular fraction. It all depends upon your method for isolation of these cells. All right, a little more work by Roger Wagner. Actually, after he left studying the fat-derived endothelial cells, he did a lot of work in parasites. Figure 7 from his publication on parasites has been shown in so many different seminars by people that where I've attended, and they're saying this is an example of a parasite on endothelial cells and fat. No, it's a picture of a parasite on endothelial cells from the swim bladder of the American eel, which is what Roger worked on. There's only one paper that I've seen where they actually cite this picture. We've utilized Roger's technique to try to identify parasites. It's a microdissection technique on in situ fat, human lipoaspirate, human surgically removed, and we've not been able to identify anything that resembles this type of parasitic investment. It's my belief, based upon my studies and other studies, that there are very few parasites in human fat. What does the primary isolate look like in following fat digestion? It looks like top left. There are a few fragments and single cells, and these are some of the other images, including Roger's images. I'm going to take you through 2 videos now. The first one is a stromal vascular fraction that has been plated onto collagen type 1. This is a gelatin-coated dish. And you'll see over about 5 days, this is after it's been washed at 24 hours. After about 5 days, you will see the morphological change in this culture. From the fragments that were there, you'll see some expansion of the cells, but cells are proliferating from all the cells in that primary isolate of the cells that have attached. And this is after about 5 days. If you take it to 7, 8 days, this is the classic image of a stromal vascular fraction primary culture. Some people will call this pre-adipocytes. Some people will call this, and based upon FACS analysis, they'll call this mesenchymal stem cells. These are predominantly endothelial cells that are beginning to lose their endothelial cell, certainly their phenotype, but they've also lost many of the markers of endothelial cells. I'll now show you the exact same isolate, but we've plated these cells onto type 4 collagen. Exact same media, exact same cells. I'm taking this from the initial plating, so you'll see a lot of cells, and then it'll be washed at 24 hours. Oops, sorry. There's the washing. Vasculogenesis takes place on type 4 collagen, and then all of a sudden, endothelial cell sprouting will take place. And this is the classic when you have a stromal vascular fraction. This is the process of in vitro angiogenesis that is taking place. I'm going to break and very quickly go through how I move from basic science studies of the stromal vascular fractions to studies of clinical application. This all began with a collaborator, Bruce Jarrell. Bruce and I were at Jefferson Medical College together. Bruce is now the president of the University of Maryland in Baltimore. But Bruce and I were studying endothelial cells in culture. He's a transplant surgeon, and he was giving me fat. He was also giving me large vessel endothelial cells. We met a patent attorney by the name of Phil Johnson, who wrote our original patents on isolating cells from fat. You see the two listed there below. Phil went on to Johnson & Johnson, and then he also wrote the 21st century patent modifications, modernization of the patent process. But he was really instrumental in writing these two patents, and one of the claims we put in included genetic modification of these endothelial cells isolated from fat. Ray Gilmartin, the president of BD, president, chairman, CEO of BD, one of his attorneys read this patent and said this could be disruptive to the sale of tuberculin syringes. Ray invited us to the new facility of Becton Dickinson at Franklin Lakes, and he internally funded the production of the original automated isolation methods. The original patent, Paul Alchez is the first inventor named on that. Joe DePisa was the main engineer at Becton Dickinson. But I have to thank Ray Gilmartin incredibly. He was one of the more unique chairman of a large biotech company that put teams together to push technology forward. And that led to this first automated system. We actually were thinking with BD and others to use cultured adult endothelial cells and not fat, develop the method for bioreactor culturing of large vessel endothelial cells, but it costs too much to do this process. So we abandoned that and turned to fat-derived cells. I show you this slide for one or two particular reasons. One, we took the stromal vascular fraction, sent it to the American Type Culture Collection for their analysis and possible distribution through ATCC of stromal vascular fraction. But they basically called me up and said, that stromal vascular fraction produces endothelial cells, adipocytes, pre-adipocytes, muscle cells, chondrocytes, osteoblasts. There are so many different stem cells in there, it's just going to confuse people. So we knew, and it was about 1985, that there were so many stem cells in the stromal vascular fraction that selling just or providing just SVF was too confusing. Joe Madry and I showed that collagen 4 could stimulate what is now known as in vitro angiogenesis of these endothelial cells. And this is used widely as a functional assessment. But we turned to utilization of fat cells, isolation of the endothelial surface from fat using the Rodbell method, placing on a graft, implantation. Here's the original method from the patent, where we mince the fat, digest it, centrifuge it, and then put it through a gradient using Percol. And that went to become the first method, took the Percol out, 1988, for treatment of a patient utilizing these adipose-derived cells. And that patient did extremely well for a year. She died of metastatic cancer, but the vascular graft that was put in formed an endothelial cell lining. And we went on to create a number of different automated instruments. The Becton Dickinson instruments that went on to clinical trials, worked a lot with the FDA to determine the good and the bad of collaginases and other things. And then Paul Kosnick from Tissue Genesis stopped by my office in Tucson, and we sat down and hatched a plan to more efficiently, using a completely automated, almost completely closed system, the isolator system that is used by Tissue Genesis. And we now have a variety of commercial systems for the isolation of stromal vascular fraction from fat that are under investigation. I'm going to finish up, Bob. Looks like I'm getting pretty close here. I'm going to finish up. All of these slides will be available in the video, but I'm going to take you through real quickly all the different variabilities that we find in the isolation of fat and how we've tried to get around some of these variabilities. First, human species variability and the effect of different things that humans do that dogs and cats and rats don't have this much variability. We're still in the early stages of trying to determine the effect of smoking and alcohol and drugs and exercise on cell yield, cell viability. We still don't fully understand this whole process. Different types of fat have different amounts of matrix associated with them. How we remove the fat, surgical excision versus liposuction. I spent a lot of time helping develop these liposuction cannulas. Here's some of my original drawings to get the fat out the best for digestion, how to digest using modified graters, cheese graters, mincing, the garlic presses. It is all over the map, the different types of mincing technologies that are developed. And I can't recommend what's the best. I can just say they're all different as far as what they provide. Collagenase, mirror, mirror on the wall. Who makes the best of all? Well, here are really the gurus of collagenase. Starts with Moses Kunitz, who is known for crystallization of proteins. Charlie Worthington worked in his lab, and then in 1947 started Worthington Biochemical. Innes Mandel was critical to purification of collagenase, and then helped understand with the Mandel assay how to measure collagenase activity. Tetsuro Kono, you need both purified collagenase and trypsin or some neutral protease to get effective digestion. Van Wart taught us that there are at least 7 to 8 different types of collagenase in the collagenase family. And then Bob McCarthy, Francis at Boehringer Mannheim, and then Vitacyte have been critical in assays of collagenase purification, recombinant enzymes. And again, you're going to be able to go back and take a look at this. Clostridium histolyticum, it all started in the 1500s with the identification of gas gangrene. One key step in this that everybody needs to know, the U.S. Army actually tried to utilize serum to block the effects of collagenase and found that serum doesn't work. Serum does not stop collagenase activity. So that's one that you can take out of your methods. And then commercialization, all these different companies sprang out of the original work that started with Charlie Worthington at Worthington Biochemicals. Kono's work, you need both trypsin and collagenase to have an effective digestion. Source of enzymes started in 1947, Worthington. Here are all the manufacturers who can provide you enzymes for the digestion of fat, and how they were derived, and how their names were changed. Worthington's name has stayed the same. Sigma came along. They had one marketing advantage, that their manufacturing facility was next to a post office, so they got their product out much more rapidly. You can buy it off of Amazon if you need collagenase off of Amazon. And I think I'm going to finish up here, Bob, because I'm just about over my time. Here's the evolution of the collagenase nomenclature from 1953, where the Mandel 40% ammonium sulfate, which turned into Worthington type 1 in the 60s. Now we're in 2024, and if you go to the catalogs of all of the enzyme distributors, here are all the different enzymes that you can buy. So I will not be able to get to this during this talk, but I'll come back. There are different types of collagenases that are available. I'm going to jump through this lot testing and end with this slide. If you want optimal stromal vascular fraction isolation, my recommendation is you can do lot testing on crude collagenase. I've done a lot of lot testing over the years. It takes a lot of people, and once you do and spend all that time doing it, you tend to want to buy up that entire lot and use it over a long period of time. Or you can go to enriched, purified class 1 and 2 collagenases. If you do that, use the Worthington level of collagenase activity, and use make sure that it has a higher concentration of protease. Type 1 Worthington is sort of the model that I use. If it's recombinant, make sure that you have a balance of class 1 and class 2 collagenases. They have different mechanisms of collagen degradation and a high concentration of recombinant protease, and digestion more than 20 minutes. If you want fragments, lower concentration of the proteases, lot test for that. And if you're using recombinant, again, low concentration of the proteases, and digestion time less than 20 minutes. Bob, I think I'm going to stop there, and I will jump, take any questions that come up, and I will jump to my final slide here with my contact information. You see where I'm going to be going in the future on this whole thing. I do want to talk about stem cells at a future time. I truly believe that ISCT and IFATs, and I was asked to be on that original ISCT group, and this is in all deference to Arnie Kaplan, but Arnie and I had many discussions about this. MSC as a designation of a cell is somewhat of a heuristic approach to this whole thing. I think if you drive your isolation methods purely to this ISCT, CD markers that are positive, CD markers are negative, you're going to throw a lot of the stem cells, the active stem cells, out to get that designation and to get that characterization that fits our definition of M cells. And I think even Arnie, if he was still with us, would agree with that. There is an incredible stem cell population in the original isolate of stromal vascular fraction that people should spend more time focusing on. Let me stop there because we're just about out of time, and take any questions that might come up, Bob.
Well, I can start just a comment to kick it off. I mean, there's a lot of different collagenase suppliers, but there's really only 4 manufacturers who are primary manufacturers of collagenase. Of course, it started Worthington. My guess is that Worthington started OEMing their products or private selling for private label to Sigma. Sigma saw this was a good business. Sigma then started manufacturing that product line. And it's interesting, all the initial numbers that Worthington did was based on biochemical characteristics. Sigma changed the game, and they used, instead of Arabic numerals, they used Roman numerals.
Right.
And they ended up actually basing it on whether it released cells. The problem with that is the biochemical aspects of these products are all over the map, especially type 11s.
And Bob, for everyone, what they did, my understanding, and I did visit them a couple of times and saw this process, they would take animal tissue of different, rat fat,
Right.
and liver and muscle, put it in different types of enzyme, and that enzyme that was best at dissociating the cells, they would then designate it type 1, 2, 3, and 4 type collagenase. So people who are using Worthington type 1 and then say, well, I'll use Sigma type 1, it's a different enzyme.
Maybe different, that's right. So then Nordmark came into the play because literally the other side happened was wound debridement, which funded all of Ines Mendel's studies because they wanted to start treating soldiers. And Nordmark just followed what Biospecific Technologies, a company that doesn't exist anymore, but just made collagenase for wound debridement. And they got into the tissue dissociation business around 2004.
Yeah, and just to interrupt on that for everyone, Ines Mandel was a PhD, but she was a professor of surgery at Columbia. So, and I talked to her about that because I moved from physiology at Jefferson to surgery at Jefferson, a different mindset of investigators and a different direction for my research. I was now most interested in how to get these cells ready for surgical applications versus my physiology time where I was trying to study basic function of those cells. So Ines did the same thing, and wound debridement was a big thing going on in her laboratory as well as other investigators at Columbia.
Yeah, and then of course Nordmark, and then basically it was Roche who really didn't make any they made a few collagenases. The OEM from Worthington's, their main collagenases, they made a few of their own, collagenase H and collagenase P, and it was the story that most of you know. It was the use of collagenase P in human islet isolation, and that variability had led to internal funding of the project in Indianapolis that we started. So Vitacyte was created. They stopped all R&D in the U.S. and we got SBIR grants. So yeah, that's kind of a history. But all those suppliers you see, if you want to see if there are Worthingtons, if they get their product from Worthington, just look at the certificates of analysis, and you'll usually find out that the assays are the same for the Worthington products as whatever private label you have. And I think 30% of Worthington's business is from private label. So I was, when we first started talking about this, Stu, what I was amazed at is how relevant your discussion is to prior art if you do in patents. Because as you very nicely show, it's very hard to invent something new because they're all incremental improvements.
Yeah. And part of this also, Bob, is, again, and I went through that briefly, on the intellectual property side, these patent examiners use very similar methods that investigators use in order to search the literature. So the keywords that they put in to determine prior art, as well as we as investigators, the keywords we put in, if you put in adipose stromal vascular fraction, that will only be a small subset of the cells that are derived by collagenase digestion of fat. If you put in pre-adipocytes and adipose and collagenase, there'll be a whole nother set of papers that come out. If you put stem cells and adipose and collagenase, it's a whole nother set of papers. So the patent examiners, and I've been on the prosecuting patent side and trying to defend patents. I will go back and take a look at the keywords that the patent examiners used in order to develop it. That indexing in keywords, which is often based upon what investigators put into the keywords for their papers, really limit how the patent office goes and identifies what the prior art is. As inventors, we are required to list all the prior art. So this is that we know of. So this is one where some people may not like me to say this, but we really should go back, and it all starts with Marty Rodbell.
Yes.
And then all of those initial papers on pre-adipocytes and, the work in the 80s where the stromal vascular fraction, these cells were starting to differentiate. That's when it all really occurred. But the patent examiners with incorrect keywords and investigators with incorrect indexing and keywords won't pick up on the breadth of all that technology. I think it's going to be real interesting when AI comes into the process of searching for the derivation of technology by the patent office, when they use Google Labs methods and other methods to go and truly search it. I'm doing it myself for the stem cell world right now, fat-derived stem cells, and I'm already picking up. There's a paper by Ray Parker from 1933 where he shows Ray Parker developed Media 199, which was used polio vaccine, and I used it in a lot of my original work. But in 1933, he discovered that if you take blood and you clot it and you put it in culture, a whole group of blood vessels forms inside that fibrin clot. That was the origins of in vitro angiogenesis. 1933. Mm-hmm. That's the last time anybody read a paper from 1933. I mean, my graduate students will only go back 4 years in the literature. But it's all, they're little bits. The pandemic actually helped because we all had to sit around and do literature searches and write review papers. Right. They shut our labs down for a while.
So the other thing you mentioned, that this can go from short-term to even 24-hour digestions. And when we've looked at this, because it's a similarity, I mean, there are people who have really low collagenase in materials, especially in therapeutic applications, we literally have to do a 24-hour Mandel assay. And so I'm assuming that the timing really gets into the amount of collagenase and the quality of collagenase. It gets into the being misled by measuring a peptidase activity. You know what I'm saying? Yeah. Instead of the true McCoy, which is the collagen degradation activity.
So what I'm picking up in reviewing that literature, and going all the way out to 24 hours, it's the understanding that as these two classes of collagenase, class 1 and class 2, one of them begins digestion from the end of the collagen fibrils. The others is a endo. It starts digestion in the middle of it. How that process progresses is important. Collagenase needs to bind to collagen in order for it to be active. So to get complete digestion of the fat, there are some investigators who have extended it using lower concentration to allow the collagenase to truly diffuse into a very small space to get the full digestion of the fat. The other one that is critical to this whole isolation method is when you do the digestion of the fat, the viscosity of that digest will determine the deposition of the stromal vascular fraction cells. If you use a very high concentration of fat to collagenase, the digest will have a much higher viscosity. When you spin it, a lot of the endothelial cells and less dense cells will stay up in the solution, will actually stay up in the fat. So that 24-hour digestion was low concentration and a higher volume of fluid to fat. The viscosity is much lower. They end up with a higher yield, probably not due to that long-term digestion, but the lower viscosity of the digest. That's one of several keys in this variation that I think will improve not only the number of cells, but you'll get far more vascular endothelium in the pellet. And by that, based upon my model, you'll get far more stem cells in the SVF pellet because there'll be more of the endothelial cell stem cell population. I don't believe it's parasites. I believe it's the endothelial cells and those original cells that are sprouting off the endothelial cells during the angiogenic process.
So can you comment on the tip cells? What are they? Because this is the first time I've ever heard of it.
Yeah, so tips in the angiogenic process, and this goes back to work by a husband-wife team at the University of Pennsylvania. Their last name is Cook, and they put little rabbit ear chambers, little chambers and big rabbits in the ear, and then they followed the angiogenic process as new blood vessels grew into that ear chamber. And then what they saw is at the tips of those vessels migrating in, so angiogenesis, new vessels sprout off of existing vessels. At those tips, that's where they saw lipid accumulation in cells. This is all morphological studies. It is all what some people call observational. They use a negative connotation to observational. But what they're seeing is those tip cells are the origins of the lipid-containing cells that then turn into adipocytes. So when a patient injects themselves with insulin at the same spot, that insulin will lead to an angiogenic process and then a steatosis, little fat deposits. Diabetics know you shouldn't inject the insulin over and over again in the same area because you will develop fat. And islet transplantation, you will see fat development in the area where the islets go into the liver. They embed. There is a stimulation of the vasculature there due to those islets and the release of the insulin. That will lead to new fat development, most likely from the endothelial cells that are present in and around both the liver as well as the islets. So that's the model. Maybe wrong, but that's the model that I stay with. It's those cells in the tips that are the true stem cells in fat.
Okay, well, Stu.
Yeah, Bob, there's my contact information up.
Yeah,
So
please feel free. Like I encourage you, one of the reasons we're doing this is we're trying to really, in this special topics and cell isolation series, really start to drill down. And I think Stu, you did a magnificent job because we do the same thing with collagenase. We know the whole history of how this whole field developed. And it's interesting to find out the way things are, as you once had on. You didn't show that slide, the slide from. Let's see if I find it. The person who was at the computer programmer. The worst words in the English language is we've always done it that way.
We've always done it this way. Yeah, the number of graduate students, postdocs, technicians who would call me, and first they'd say, "Don't tell my advisor that I'm calling you." But I read your paper. Can you help? And I always ask, "Well, how are you doing the isolation?" And they say, "Well, I'm doing it the way I've been told to do it." It's the way we've always done it, but I think there might be a better way. Can you help with this? So
That's right.
yeah, that's the I forget her name. She was an admiral.
Yeah.
Cobalt. She developed.
She invented cobalt. Yeah, but anyways, the long and short
Always
is
done it this way. Yep.
Yeah, I mean that's what we see all the time, and you know, a good example is islet isolase used the Ricordi procedure, and it may not be the best procedure.
Yeah, but not to get any quality people who are on the line upset, but man, once you get all your SOPs in place and your BPRs, batch production records are all in place, to make a change in the process.
Yes, yes.
Oh,
that's absolutely true.
God.
My God, yes, yes.
Yeah,
and that's the other thing. And just go on a little sidelight is that we're going to be working on doing an educational thing for the new ancillary materials standards that ISO published. And the biggest dilemma we have is, how do you know the specifications of your enzymes?
That's right.
And I'll be honest with you, no one does.
No one does. No, we use Wunsch assay just because.
Right.
Yeah, we talked to FDA. We talked to lawyers about collagenase, and it could be lawyers defending a company against the FDA, what collagenase is. And, oh, yeah, we analyze it using the Wunsch assay. You know, it gives us a little bit of an idea of class 2 collagenase activity, but otherwise the total protease mixture in there doesn't tell us anything.
Right.
So that keeps the lawyers happy, FDA, and a few other people happy, but doesn't give us the answer that we really need.
Right. All right. Again, thank you very much, Stu, and thank you all for participating in the webinar. Fill out the polls. I hope you have when you've seen them. And what we'll do is, and feel free to contact Stu if you have any questions about his presentation. And thank you again, and hopefully we can arrange for you to come back in next year to give the part 2 of the presentation.
All right, happy to do it.
Good. Thank you.
Take care. Bye, everybody.
Bye.